All right. Hi, everyone. I'm Sriram Sankararaman. I'm a faculty here at UCLA. I'd like to start by apologizing. I missed most of this week's short program, just recovering from COVID, but I'm glad that I could at least be here for the final day. So my group is broadly interested in machine learning applied to different types of genomic and biomedical data analyses. So we are very interested in using machine learning to understand population genetics and evolutionary processes. We're also interested in understanding clinical data and how that can be used, machine learning can be used to improve clinical decision making. And the other important area that we think a lot about is complex traits and the link between our genomes and complex traits. And so today's talk is primarily focused on this area. And a couple of reasons why I like to talk about this. The first reason is I think there are some really interesting challenges here, methodological challenges, really at the intersection of statistics and computer science. And so that makes it extremely exciting and interesting. The other reason I like to talk about this is some of this work came directly out of conversations that happened in previous CGSIs and this other meeting that happened at Bertinoro. So definitely it's a great value to have these coffee breaks and these extended conversations, and hopefully I'll give you an update on some of these projects. So my interest in thinking about complex traits has been really spurred by the growth of these biobanks, and I believe there were talks about this over the course of this week. So biobanks are essentially these repositories that combine phenotypic information, rich phenotypic information with genetic data, often in the context of health systems. So a very prominent example is the UK Biobank, but then there are other such efforts ongoing. So there's things in Japan, in Finland, and in different places in the US. And one of the things that's happening in the context of these biobanks is scale. So we are now at a stage where many of these biobanks are collecting genetic and phenotypic information across tens, hundreds of thousands, even millions of individuals. So the question that I'm interested in is how can we learn about biology, about complex traits from the millions of genomes that are being accumulated in these biobanks. So that's a broad set of questions, and there are many different angles to it. There are statistical aspects. So how do we actually deal with the complexities of the data that is being accumulated here? How do we model the data accurately? There are computational questions about scaling, about getting inference algorithms that can scale to these large data sets. There are interesting questions of privacy. Different biobanks have different restrictions on how they can share their data. And finally, there's issues of interpretability. So often you're running these methods, they are complicated models, so how do you actually make sense of some of their outputs? For today's talk, I'm going to primarily focus on the statistical and computational aspects of learning from biobank-scale data. And just to make things concrete, you've probably seen this. So the kind of data we are going to be dealing with is genotype data from a set of individuals coupled with their phenotype. So you have this matrix. Every row here is an individual. Every column represents a position in their genome where there is variation. So we are going to be focused on a class of regions which are single nucleotide polymorphisms or SNPs, where there is variation at a single character across these individuals. And then we have phenotypes that are linking the genetics to the trait of interest. And because of the way mutations, of the fact that mutation rates tend to be low, in most places you only have one or the other possible mutation at a given position. So we can essentially represent these SNPs by this ternary matrix, 0, 1, or 2, which basically encodes how many alleles, how many mutations an individual carries. So 0 means they don't carry any of the mutations, 1 means they carry 1 copy, 2 means they carry 2. All right, so we are given this matrix of genotype and phenotype, and now let's think about some key inference problems that we'd like to be able to perform. So one key inference problem is that of population structure. So looking at this genotype matrix, we'd like to be able to understand how these individuals relate to each other and to their populations they might arise from. Another important question is genetic architecture of complex traits. And finally, in the context of these biobanks, there's often issues with missingness. So you might think that you've collected data on 1 million individuals, but in reality, for many different reasons, you've actually only got a fraction of this data. So there's a lot of missingness that's happening. So I won't talk about population structure. So this is something I've talked about in previous CGSIs. Instead, in today's talk, I'll talk about genetic architecture and dealing with missingness. So what do we mean by genetic architecture? So broadly, we want to be able to understand this function that maps a person's genotype and environmental measurements to their phenotype. So one specific way of quantifying this is this notion of heritability. So heritability is essentially a measure of how much genetic signal a given phenotype has. You can think of it as how predictive a phenotype is from genetics, or what proportion of the phenotypic variation is explained by genetics. So as I said, it's a measure of genetic signal. For those of you who don't think about genetic data, this is actually a broadly applicable idea. So you might be working with epigenomic data, with image, with methylation data, and you can ask how much signal arises from this data with respect to some phenotype of interest. And so heritability is basically this measure of signal associated with some set of features. So how do we think about heritability formally? So we have phenotypes, we have genotypes, and we are going to posit a model that connects genotype to phenotype. So we'll start with a linear model. It's a simple model, but it already has some interesting issues. So we're going to write our phenotype as a linear combination of each of the SNPs in our genotype, where these betas are the effects of these SNPs. And then everything else that cannot be explained comes from the environment. So now you have a linear model. In principle, you can fit this model. You can try to estimate the effect sizes. In practice, it's hard because even with 1 million individuals, the number of SNPs that you're dealing with tends to be much larger. So you're in this high dimensional setting where the number of variables, the number of SNPs, is much larger than the number of samples or individuals. So we can't estimate these accurately, individually. So to be able to understand about genetic signal, we are going to put some assumptions. So a common assumption is to assume that these effect sizes come from some distribution. For example, they might come from a Gaussian distribution with some mean 0 and some variance. And we term this variance associated with the effect sizes of these SNPs as the genetic variance component. Likewise, our environments, these are random variables coming from a distribution with some mean 0 and some other variance. We term that the environmental variance component. So given this genetic variance component and the environmental variance component, we can estimate this quantity called heritability. So it's a number between 0 and 1. Values of heritability close to 0 means the trait is essentially not predictive by genetics. Values close to 1 means it's highly predictive from genetics. So this is a very simple model. It has these 2 parameters we'd like to be able to estimate, but in practice you can extend this in lots of different ways. So one way to extend this is to say it's not realistic to assume that every SNP at every position in the genome comes from the same distribution. So you can say maybe if a SNP falls within a gene, it comes from a particular distribution, and if it falls outside, it comes from a different distribution. So now we are introducing additional genetic variance components. And now you can ask how much genetic signal, how much heritability is attributable to SNPs that come within genes from outside. So this type of analysis has been extremely influential in trying to localize where genetic signal comes from. So there, for example, this early paper showed that there was an enrichment of genetic signal from particular regions of the genome. So this variance components model, this class of models that we're talking about, can be made quite general. So here's the basic setup we are interested in. So we have our phenotype. We are dividing up our genome into k bins or components. So xk is the genotype that has been assigned to the kth component. And there are effects associated with the SNPs that fall within that component. So that's beta k, and then everything else is environment. And so all of the effects that fall within one component, they're going to be assumed to come from a specific distribution which has a shared variance component. And likewise, you have the environment which comes with its own variance component. So we have a classical statistical model, and our goal is to estimate these k genetic variance components and the environmental variance component. So we know how to solve this. It's a statistical model. we can write down the likelihood and maximize. Now the challenge happens because of the scale that we are thinking about. So when we think about applying these kinds of approaches in the context of biobank-scale data, maximum likelihood approaches are computationally expensive. So typically they're going to scale as cubic in our sample size. There's been plenty of interesting algorithmic work to actually make these algorithms scale, but it's still challenging to apply to biobank-scale data. Yeah.
So you write that you maximize and it is n cube. This is the standard linear maximum likelihood.
Yes, so we are in a linear mixed model setting. So y is a linear function of x plus some noise, and now you write down the likelihood. So even the simplest model is actually challenging to fit at scale.
Still, it is computationally efficient. It is not hard. Yeah. Okay.
It scales cubically in the sample size. So imagine your sample size is 1 million people, then you have challenges in estimating maximum likelihood. Questions? Maybe I'll pause here if there are questions before we move ahead. Okay, so when we started thinking about this, we said, okay, in basic statistics, there are these two classes of doing estimation. One is maximum likelihood. So there's another classical approach which is based on moments, so moments of the model. And see, so these are called method of moments approaches. So the idea is you don't look at the likelihood, you look at specific moments, could be the mean, could be the covariance, and you try to find values of your parameters so that the theoretical moments match the sample moments that you have estimated from data. So these two approaches have trade-offs. So maximum likelihood tends to be very efficient. You get very precise estimates, provided your model is correct. On the other hand, when your model is incorrect, it might be advantageous to simply focus on a few moments and get them right, rather than focus on the likelihood itself. This is a classic approach, and in genetics, it has been proposed and goes under the name of Huysman-Elston regression, HE-regression. So now let's see what happens if you try to apply this approach in the setting. So the way our model has been framed, the first moment is 0, so there's no information there. So let's move on to the second moment. So we can look at the covariance of our phenotype. And it turns out you can write down the covariance of this phenotype in this model. It is a linear function of our variance components, these parameters. And the coefficients are given by these matrices. So the covariance is a matrix, so you have coefficients which are matrices. And there is one coefficient for every one of our components. Turns out this matrix is an n by n matrix, and it goes by the name of the genetic relationship matrix. And it basically tells you what is the relationship between every pair of individuals at those set of SNPs that you're considering. So that's the theoretical moment, and then we have the sample moment, which we can estimate from the data. And now the goal is to be able to find values of our variance components so that these two quantities are close to each other. So that's the setup. It has some advantages. So beyond the statistical advantage, it turns out for this model, there's a closed form solution. So unlike maximum likelihood, where you have to do numerical optimization, method of moments allows you to do closed form optimization. So you can write down the solution. We are not done yet. You still have a computational problem, and the computational problem arises from the fact that these genetic relationship matrices still need to be computed. So this is an n by n matrix, so if you actually try to compute it, it's still going to scale as quadratic in your sample size with a fairly big constant, which is the number of SNPs. So we're still not at a place where we have a scalable solution. So our thinking here was, if you look at the form of this solution that you get from solving the method of moments equations, it turns out you don't actually need to compute this n by n matrix. So you have a linear system, and the coefficients of the linear system only depend on traces of this matrix or traces of products of this GRM. So essentially you're doing all of this work, computing this large matrix and then shrinking it into these small numbers, these traces. So this gives us an approach to avoid computing the GRM, and that comes from the fact that traces of matrices can be estimated by these randomized estimators. So this is again classical linear algebra going to the 80s, and the idea is if you basically multiply your input matrix by a random vector. It could be any distribution as long as it's mean 0 and identity covariance. And then you average several of these, then this gives you an estimator of the trace. So you're multiplying essentially the genotype matrix with these B random vectors. So another way of thinking about this, for those of you from a more computational background, is we are working with essentially a sketch of the genotype matrix. So this genotype matrix is huge. You multiply it with some number of random vectors, and that reduces its size. So this is a parameter, the number of random vectors B, that you need to multiply this with. So the efficiency of this approach depends on how many random vectors you need. Smaller, the more computationally efficient. And in fact, it can actually scale as sublinear in the size of this matrix. So the reason why this happens is this matrix is a special matrix. It's a genotype matrix. It's 0, 1, and 2. So because of this structure, you can actually do matrix multiplication extremely efficiently. However, the key question is, what do you lose in terms of statistical efficiency? And what is interesting, and I'll show you in a bit, is this method is accurate for even small values of B. So B could be as small as 10.
Sorry, N is sample size?
Yeah, N is sample size, M is the number of SNPs. So UK Biobank, N is 500,000, M is million, 10 million, and B is 10. So clearly there is a huge savings there.
And K, you have K there. In the previous slide, you have K.
K is the number of components. So you're taking your genotypes, your SNPs, and you could partition them into K. That makes your model more expressive, but it doesn't particularly change the computation because you're just dividing up M into smaller numbers. Yeah.
And B is the sample size, the number of samples, B.
B is a parameter. So when you're designing this algorithm, you need to figure out how many random vectors you're going to use to summarize your data. So this is a design decision.
Yes.
And of course, we'd like to make it as small as possible without losing statistical efficiency. Yeah.
So you're effectively sort of randomly projecting your matrix down. How about dimensionality reducing your matrix?
Yeah, so there are different choices. So we'll come to that in just a bit. So yes, so here the simplest approach is think of these random vectors as IID Gaussian. That works okay. But there's a whole set of design choices you can make here about choosing different distributions, different approaches, which might give you better or worse results. But the simplest approach actually works quite well. This is Johnson-Lindenstraut's, but it's actually important here. It's there is issues about why it works as well as it does, and it's not completely obvious. So I'll come back to that in a bit. Okay, yeah. So the question is, in our choice of the random vector, does it have to have certain properties? Yeah, so for this to hold, for you to get an unbiased estimator of these traces, which is the quantity we care about, the only thing that you need is 0 mean identity covariance. That's all you need. So it could be anything. For example, it could be Gaussian, it could be Rademacher, so maybe you can get discrete random variables, and there are other potential choices as well. So in all of these results, we use the simplest, but there's a whole design decision which we have not explored at all. So there's another point which is important with this method, and it turns out it's a closed form approach, and you're working with a sketch, so it's also what we call a streaming approach, which means you'd never have to read the full genotype matrix into memory at once. You could actually read it in one pass or two passes and compute, which also means there's a big memory savings here. So this is important when you're trying to run these on large data sets. Okay, so let's look at some results. So we call this randomized Huysman-Elston. So it's a randomized version of the Huysman-Elston regression. So here are some simulations. These are, I won't spend too much time on the simulations. This is all published, but I'm happy to also talk about this if there are questions. So here we're asking how much do you lose relative to maximum likelihood. So remember, maximum likelihood under the true model is optimal. So here we did a bunch of different simulations, and the goal is to estimate the heritability of the phenotype under these simulations. And as you can see, we have different types of simulations, different genetic architectures, and we are looking at these heritability estimates coming from maximum likelihood, in this case GCTA, to what we have from randomized HE-regression. The standard errors are bigger, but not by much. It's about 10% larger. So you don't lose much with even 10 random vectors. So then the additional side of this is you can now scale this approach. So you can apply it on biobank-scale data. And for that type of data, it's hard to compare it directly to maximum likelihood approaches. Instead, in human genetics, there's been a lot of work in trying to run these methods using summary statistic approaches. So how do you summarize your genetic data, your summaries, your GWAS data in different ways so that you capture most of the information there? So these are comparisons to these different summary statistic based approaches. And what you see across these simulations that we ran at scale is, in terms of the relative bias of these estimates, which is one metric we looked at, these randomized Huysman-Elston approaches are actually quite a bit better than many of these summary statistic approaches. I must mention there's a gold standard here, the GRE method. This is from Bogdan Pisanuc's lab, and this is a fixed effects model. It's a slightly different approach, which is provably unbiased. So RHE is actually doing quite well relative to that. And now this is looking at the scalability. So as you increase the sample size, we find that the RHE approach scales much better than the maximum likelihood approaches. And it's not just about sample size, it can also do computations across different annotations and different ways of partitioning the genome. So you might not just care about heritability as one number, you might want to look at heritability in genes versus outside, in regulatory regions versus outside, and it can actually do this across large numbers of these annotations. Yeah.
So how does this method compare if instead of doing this sketching approach, you read the data in batch and update until you reach the end of the batch? So basically, your HE is closed form. Instead of reading the entire data, you can read the batch, update your estimate, which is closed form, and then go until you reach the end of the batch.
I think math. Okay, that's interesting. So there are some issues though, and the issue is this approach. When you're reading it in batch, there is still interactions in your estimates between data point 1 and data point 1000. And so the randomization is preserving that. Whereas if you just read it in batch, there's a question of how do you get that interactions? And so maybe there are approaches. So this is almost like doing an SGD on the objective function. So there might be some alternate ways of thinking about it. Okay, so we have a method that we can now apply to UK Biobank. So here is an exemplar setting. So unrelated white British individuals, 300,000 of them, 8 million SNPs. So we are looking at different types of SNPs. We have SNPs that are present on the array, relatively common, minor allele frequency is greater than 1%. We have imputed data, and again, MAF greater than 1%, and imputed data going down to 0.1%. And here what we are estimating is the heritability, the total heritability for these SNPs. And what you see here is across all of these traits, there's an increase in heritability as you go to rarer SNPs. This is not something that this is something that has been observed previously. And how much heritability you gain depends on the type of traits. Across different traits, it's about 10% increase in going from array to low frequency imputed. And for traits like height and BMI, there's quite a bit in terms of additional heritability that's gained. As I said, we don't only have to look at this genome-wide number, this genome-wide heritability. Often we are interested in figuring out where heritability is focused. So here now we can partition heritability across different functional genomic annotations. So these are annotations like whether a SNP lies in a coding region, whether it's conserved, whether it lies in enhancers, and so on. And so now when we run this, we find that there is, for example, enrichment of heritability. So this is a measure of how much heritability you expect versus how much you actually observe. So high values mean there is more heritability than you expect. And so you find that if, for example, in conserved regions, there is enrichment of heritability across all traits. This has been previously observed in the literature. Whereas when you look at enhancers, there is more trait-specific enrichments. For example, diseases like asthma and eczema tend to have trait-specific enrichment in enhancers. Okay, so this was kind of a warm-up. We started out with a linear model. and showed that you can actually fit these linear mixed models at scale, and they seem to actually be quite accurate. But now this led us to think, what can we do? Can we go beyond these linear models? So what about nonlinear effects? We now have data sets where you can potentially have the power to see these effects. So can we estimate these in large-scale data? And kind of the key takeaway is this approach, this randomized method of moments, can be extended to each of these settings. Yeah.
Sorry, I'm just asking a very basic question. I don't work in this field, but so when you say it has the power to estimate nonlinear effects, it's presumably some kind of interaction effect. And what do you mean by power?
Yeah. So in human genetics, there's been a lot of work in trying to understand different types of nonlinearities, gene-gene interactions, gene-environment interactions, dominance, and all of that. And typically, when people have done calculations and have run these methods, there's been very limited evidence for these kinds of effects. And one hypothesis is it's a sample size issue. Once you have these large enough sample sizes, you might be powered to see some of these. So that's one aspect. The second aspect is, of course, the multiple testing burden. For example, if you're trying to test every possible pairwise interaction, then how do you deal with that? So there are both of those that are going to come into the picture, and we'll see how thinking of it in the context of a variance components model can help with some of those. Okay, so let's start with one notion of nonlinearity, and this is dominance. So the idea is, if you're looking at one SNP, you're moving from having two alleles which are lowercase b to two alleles which are uppercase b. And if the effect is going to be intermediate when you have the heterozygote relative to either of these homozygotes, you're going to say it's an additive effect. And the question is whether there's a deviation from this. So we call this a dominance deviation. And so now the key idea is there dominance deviation across the genome that influences a particular trait? So here again, we're not looking for a specific SNP and asking, is there dominance here? Not even at a particular location. We're asking genome wide, is there evidence for systematic dominance effects? So the way we do this is we again extend our basic linear mixed model. So we start with this component, which is the additive variance. So this is the genotypes coded as 0, 1, 2, so an additive effect. And now we have a second component, which is encoding the dominance deviation. And so now you have effects for the additive component, and the variance component is the additive variance component, and you have effects for the dominance variance component, Now you can fit all of these together. So you can fit both the additive and the dominance variance component for given traits, and you can ask, is there evidence for dominance variance? So nothing changes mathematically. It turns out you can now apply these approaches as is. Yeah.
Question. So your dominance variance is independent of the additive variance. Like sometimes we kind of assume that if there's a SNP that's like have larger variance in the there's no interaction. It does not really like if you think about every SNP. And if one SNP have actually larger beta, I feel like the gamma should also should be larger instead of independent.
Yeah, I mean there are two issues here. One is the encoding. So I haven't got it into the details. There are different ways of encoding the dominance effect, the D matrix, and some ways of encoding are actually going to make it hard to estimate. So they're not going to be orthogonal. So you need to find an encoding, and this is work that people have thought about, which is how do you encode the D matrix so that it's orthogonal to the additive matrix. So you're actually getting estimates of dominance which are not captured by the additive component. There are other issues here, which is these dominance effects, for example, they can interact, especially if you imagine across allele frequency spectra. You might imagine that rare alleles are more likely to be dominant than common ones. That's not something we've explored in this work, but you could again imagine going back and say doing a partitioning and trying to see if there is evidence for dominance across allele frequency spectra. But yes, this is a more basic model. So, so it turns out you can fit this model again to the UK Biobank, and we did this. We looked at 50 quantitative traits, and we've estimated the additive heritability across these traits. This is a set of traits that includes height, BMI, so anthropometric traits, blood biochemistry, and so on. And generally, you find that there is substantial evidence for additive heritability. This has been seen before, of the order of 20% across these traits. But there is very little evidence for this dominance deviation effects. So in fact, when you do a formal test, there is no evidence of dominance deviation. No trait has any evidence of dominance deviation being greater than 0. So from the largest analyses that we could do, we conclude that there isn't any, if there are these dominance deviance effects, they are going to be fairly small. We can look at another type of nonlinearity. So this is interactions from genetics and environment. And this is possible because in these biobanks, you're not just picking up one phenotype, you're also collecting other phenotypes, which could be modulating your genetic effects. So the basic idea of gene-environment interactions is you have some trait of interest like BMI, and you have a person's smoking status. And depending on the smoking status, a particular genetic variant might have different effects on that phenotype. So again, the question we are interested in is not in identifying individual genetic variants. There's been some work that has been done there. But in trying to quantify genome-wide, is there evidence for gene-environment interactions making a contribution to a complex trait? So this can again be done in this variance components framework. So now we have our additive component. We have another component, which is the G by E component. And all that is taking your genotype and some environment, any environmental variable, and you now form a product. So it's essentially a Kronecker product of the environment with every one of your SNPs. And there is an effect associated with that. And now you're asking, what is the variance component associated with those effects? So that's a G by E variance component. So now you have an additive and a G by E variance component. Here, there's actually more technical challenges. So even a method which is linear, a method that was linear for fitting additive effects is not sufficient to be able to fit G by E. And the reason is now you're forming this matrix which is taking all your SNPs and taking the product with all the environments, and you cannot actually have both of those and operate on them at the same time. So there is additional work in terms of how you choose these random vectors to allow you to avoid those computations. I can talk about that later if there's interest. There's another kind of technical challenge in fitting and searching for these G by E. It turns out oftentimes there is differences in the variance across different environments for a phenotype. So this is some heteroskedasticity in the variance across different environments. And if you don't model it in the model, that might be captured as G by E. So you need to have this additional term, which I haven't put here, which is a noise by environment interaction. And this is just some simulations that show that you'll have inflated estimates of G by E if you don't account for this heteroskedasticity. So now we fit this model. Again, we're looking at about 50 traits in the UK Biobank, the same 50 traits. And this is unpublished work, so this is still ongoing. We are still making sense of this. So here we first looked at one environment, which was sex, and looked for gene by sex effects on different traits. And we find fairly strong effects. For example, testosterone, waist hip ratio, body mass index have fairly strong G by sex effects. And some of these are comparable to their additive heritabilities. Here's another example. We have medication information in the UK Biobank. So this is statin usage. And so now we looked at G by statin for different traits. And you find that the traits which show significant G by statin are traits like blood pressure. Then there is things like cholesterol related traits. And finally, there's also glucose and HbA1c related traits. So we're still trying to figure out what the implication of that is in terms of statin usage. Yeah.
Hey, Sriram. So how well do you think these dominance effects and G by E interactions are captured by the Gaussian distribution? Or do you think it's better to use something more like, have a stick, a zero, and then a long tail distribution?
Right. So there are several places. So first question is, yeah, that's actually a good point. So the method of moments, although I introduced it as Gaussian, is unbiased irrespective of the underlying distribution. So this is where actually the method of moments has some advantage over a maximum likelihood approach. So it could be a spike and slab, it could be heavy tailed. The estimates from the method of moments are still unbiased. So that's one thing. There's another place where the distributional assumption matters, and that's on the phenotype. And for example, you might take your phenotype and log transform it, or you might do some other transformation. And so what might appear as interactions on one scale may no longer appear as an interaction on the other scale. That's a more challenging question, and we don't have an answer to that. You have to look at it on a case-by-case basis. Yeah?
Yeah, I just wanted to follow up on that. So, for example, when you show this, the result of testosterone or West equation, do you still see them on a different scale? For example, if you were to log BMI or to log testosterone?
Yeah, that's a good point. We're still looking at it in different ways. Yeah, there is a question of what good scales are. So we have tried a few of them. For example, there's even questions of how do you adjust for, what are the other covariates that you should be adjusting for. For example, when you're looking at waist-hip ratio, do you adjust for BMI? What the implications of that is. So yes, these results, they could change depending on the scale. So we still haven't figured out what the appropriate scale to be looking at for those are. Yeah. But happy to hear any thoughts about any of those. Okay, so finally, you can look at G by G, so gene interactions. So now what we have done is we have replaced our environment by genetics. So the challenge, as we said, is now when you start looking at gene-gene interactions, you have many different pairs that you're going to be testing. So you can still do this in this variance components model, where what we're going to do is not going to look at every pair, but look at one SNP. This will be our SNP of interest, and look at its interaction with all the other SNPs in the genome. And what we're interested in asking is, does this one SNP show evidence of G by G when paired with all the other SNPs? So this is an idea that comes from Crawford et al. They call it the marginal epistasis idea. So you're not testing for every pair, you're going to one SNP, forming a test with all the other SNPs, and then doing a test of epistasis. So this brings down your multiple testing from quadratic to linear. And again, the question is, how do you do this at scale? It turns out you can use the same randomized method of moments idea to fit these G by G variance components and estimate them. So here is just some simulations. So in principle, we could go and do a scan for marginal epistasis. We could look at every SNP in the genome, try to plug it in. Our approach is still not computationally efficient for that. So instead, what we do is we take SNPs which have been found to be significant in a GWAS using a marginal GWAS test. And then we ask, is there evidence for marginal epistasis, G by G? So here are some simulations. These are simulations where there are linear effects, no epistasis. And now when you do this kind of a test, you find that it's well calibrated. So you're not going to get false positives using this kind of an approach. And then again, we applied it to our 50 traits, first doing a GWAS, picking those SNPs that are significant, doing some LD pruning, and then running it through this marginal G by G approach. It turns out across these 50 traits, there is evidence for strong, if you look at the negative log 10 p-values, marginal epistasis effects. Again, we're still in the process of understanding these. One of the challenges with G by G is there's a lot of literature that shows that what appears as interactions could actually be explained away by some type of missing data. For example, you have array SNPs, you're missing some SNPs. So what looks like G by G on array data goes away when you start getting additional SNPs. So again, because the approach is efficient, we've replicated this. So we've looked at the array data and also did the same test on imputed data. Some of the SNPs go away, but a large number of these marginal G by G effects persist. So at least some of these are likely to be real. And again, this is still ongoing work, so happy to have any comments on this. Okay, so now what about going beyond these pairwise effects? Can we think about that? One way to think about that is we replace our GRM. This GRM is basically taking two individual genotypes and computing an inner product by some nonlinear function. And this is often called the kernel function. So the kernel function takes two individuals and computes an inner product in some potentially higher dimensional feature space. So an example of such a kernel function is this RBF kernel, which has this form. And it's not obvious, but it's basically what it's doing is it's projecting your data into an infinite dimensional feature space and performing inner products there. So now can we actually try to test for these types of higher order effects? So our goal is to test whether the variance component associated with this kernel is 0. So this is a type of approach that has been made prominent by this approach called SCAT. And primarily the SCAT approach has been used for testing for linear effects. And in those settings, you can actually make this quite efficient and scale to large data sets. But if you're now trying to do this with kernels, with higher order effects, the score test, which is the test that is being used here, again scales cubically with your sample size. So the way you test this is you form a score test of the null hypothesis. It's essentially a test which takes your kernel K and computes this quadratic form with the phenotype Y. And there is asymptotic theory that shows that this score test statistic has a distribution that looks like this. It's a sum of a bunch of chi-squared variables with the weights that are given by the eigenvalues of this kernel matrix. So if you want to compute a p-value, you have to compute the eigenvalues of this kernel matrix, which scales as n cubed. So the question is, can we now try to make this scale? And the key idea, this comes from the machine learning literature on kernel machines, is for certain types of kernels, we call these shift invariant kernels. So these are kernels where the result only depends on the difference between the two inputs. So the RBF kernel is an example of a shift invariant kernel. It turns out you can approximate this kernel evaluation by sampling from its Fourier transform space. So this is a theorem that is well known in the kernel machines literature. So the idea is now you don't do a random projection in your original space, but in the Fourier transform space of a given kernel. So every kernel has a Fourier transform, and for certain kernels, we can now sample from this Fourier transform and build random projections in this space. So now the idea is we replace our original kernel with an approximate kernel, which is formed by sampling from its Fourier transform basis. And this gives us an approximate score statistic, which also has a distribution, which is a sum of chi-squareds with eigenvalues that are given by the eigenvalues of this approximate matrix. So why is this useful? It turns out now the eigenvalues of this approximate matrix can be computed in time which is linear in your sample size, and this d parameter, where the d is the number of samples that you draw from this Fourier transform. So it's the number of dimensions you're using to approximate this kernel. So provided d is small, this kind of a kernel can actually be evaluated and tested accurately. So again, we've looked at it in terms of its calibration. It turns out that this approach doesn't have any false positives when you have only linear effects and no higher order effects. When you compare it to doing an exact kernel, the power is actually quite close as long as this dimension, approximate dimension, is reasonable. So 25 to 50 is something that works well, and it scales much better than having an exact kernel. And now we've applied this again to the UK Biobank. Here, we can't apply it genome-wide. We have to look at individual windows along the genome and apply it. That's the setting where this kind of an approach can actually work. And so we've done these types of scans testing for these higher order effects after regressing out linear and pairwise effects. And we are beginning to find some evidence of specific regions of the genome which have these effects which can't be explained away by linear quadratic effects on specific traits. Okay, and this approach can also be applied across traits, multiple traits, so we can use it for genetic correlation. I'll skip this, and it's kind of a very similar type of story. And I want to get to the final part, which is about missing data in biobanks. So everything so far I've talked about assumes that you have all the traits available, measured at scale for this to be relevant. But in reality, the picture looks something like this. You're interested in some trait like alcohol addiction, and it's only observed in a fraction of the participants in the UK Biobank. So missing data is a big problem. Many of the traits of interest, of clinical relevance are missing. And some of this is cost, logistical issues in acquiring them. So being able to impute or fill in missing data is a major challenge. This other challenge is the data is not missing at random. So often, for example, they'll ask 10 questions of an individual, and whether they answer the second question depends on whether they've answered the first one. So there is structure in this missingness that needs to be accounted for. So when we started thinking about this, we said, okay, can we come up with a method which is able to look at correlations across traits to do imputation? And beyond just looking at correlations, maybe there are nonlinear effects that matter here. So one way to think of this is to think of an autoencoder, where you have your original data that gets mapped to some hidden layer, and that gets mapped back to the original layer. Now we can repurpose this for imputation by masking some of these entries and seeing how well it gets reconstructed, and using that in our loss function. So trying to learn a model which reconstructs the masked entries. So the challenge is in figuring out these masked entries which actually reflect the missingness patterns in the data. As I said, missingness is not random. So we have this approach where we are doing masking, which is essentially copying the missingness patterns that are present in the data into new entries, which means, for example, if there are two phenotypes that are missing in the same individual, we would mask those two entries in another individual and ask the autoencoder to reconstruct it. So we term this a copy masking approach, which essentially propagates missingness patterns that are seen in the real data. So this approach, which we call autocomplete, we then compare it to other approaches for imputation. So one approach which is actually does really well is soft impute. So soft impute you can think of as a linear version of this model. So it essentially projects to a low dimensional space and then reconstructs. And actually, a soft impute does really well for the different types of traits we've looked at. One was a cardiometabolic, another one was a psychiatric disorder. However, having nonlinearities does tend to improve imputation quality. We also compare to other approaches. For example, there are these deep learning approaches that have been proposed in the literature. However, it turns out that they are challenging to learn at scale, and so they don't do quite as well as the simple autoencoder. Yeah.
But this is essentially a sequence, right? Have you used transformer-based models?
So these are phenotypes. So there isn't an inherent sequential structure. But of course, there is some kind of correlation. So there could be local sequences, for example. So transformers could work, or looking at graph neural nets could also work. So this was our kind of first attempt at seeing whether any type of nonlinearity can actually help with imputation here. And here is kind of a closer look at pairs of phenotypes comparing autocomplete with soft impute. And basically what we are seeing here is for several phenotypes, depending on the amount of missingness, there is a statistically significant improvement in the imputation accuracy, both for the cardiometabolic and the psychiatric disorder phenotypes. So now that we have these imputations, one natural question to ask is, does it help in a downstream application? For example, do you find new GWAS hits? But before you do that, we want to know that there isn't a bias associated with the imputation. So we did this experiment where we're taking a phenotype that is actually fully observed. We mask it. In this case, we mask out 2/3, and then we impute. And then you do GWAS on the imputed data, and you ask how many of those would replicate on the original observations. So it's a way of trying to convince ourselves that we're not getting non-replicable associations. So this is one phenotype that we looked at. And here in this case, when we look at all of the GWAS hits that come out of this imputation, we find that they would all have replicated in the original observed phenotype. So it is telling us that this phenotype imputation is actually doing a reasonable job.
Super interesting. Deal with some output testing, because
Yeah, I guess there's a question of, if you think of all the phenotypes together, what the appropriate multiple testing is. I guess in this setting, we are just looking at one phenotype, so it's the standard univariate multiple testing. Yeah, the question of, we haven't thought very carefully about what happens in a phenome-wide setting. And now we did this for phenotypes that are actually missing. So several of these are phenotypes that are relevant to the psychiatric disorder data set. They're highly missing, so missing in 60 to 80% and above. And unexpectedly, we find that there is new loci that we discover, which are substantially more loci that you discover by looking at the imputed data. So clearly, there's value to be gained by doing this kind of an imputation as a pre-processing step to analyzing some of these phenotypes. Okay, so with this, I'll stop here, and I've just given you a few vignettes of some of these types of issues, types of modeling, inference issues that arise when you look at biobank scale data. So I talked about variance components analyses, and for us, the biggest learning from there is the effectiveness of randomization. And I would even say it's unreasonably effective. So empirically, it seems to work. What's missing for me is an understanding of when it works. So we're missing some theory, and so I think that would be interesting to understand and figure out. And having applied it to data, what's interesting is now beginning to look at some of these nonlinear effects. So as mentioned, there's negligible evidence for dominance deviance. However, there's evidence for other types of interactions, G by E and G by G, which we are working on, and also trying to localize these signals. So all of these are genome-wide. Of course, you can ask, can you try to localize these and get a better understanding of where these signals come from? And then I mentioned the second vignette, which was phenotype imputation. And the learning there is a power in using nonlinear models, but they need to be tailored for the missingness, the type of structure that's present in the missingness. Of course, if you're able to get it to work, it does increase power. But again, we still need to understand in more detail the quality of these imputed phenotypes. For example, we've had discussions where our colleagues tell us that maybe the imputed phenotypes are better in some sense than the original phenotypes. They might be denoising some of the original phenotypes, but that's on a case-by-case basis, and we haven't yet understood it more systematically. With this, I'd like to thank all our collaborators. I'd like to also acknowledge the students who did the work, Ali, Ariel, Ulzi, and Boyang, and funding sources, and happy to take any questions.